Welcome to Biology, the scientific study of life. Experimental evidence for behavioral evolution. Looking for more direct ways to demonstrate the evolution of behavior, researchers are increasingly turning to laboratory and field experiments using organisms and short lifespans, enabling the observation of change over many generations. Laboratory studies of Drosophila foraging behavior. In a few cases, researchers have been able to link behaviors to specific genes. For instance, Marilla Sukowski of the University of Toronto studied a polymorphism in a gene for foraging in Drosophila melanogaster called 4. One allele 4S results in a behavior phenotype called sitter, in which the fly larva moves less than average. A different allele for R to the power R results in a rover behavioral phenotype in which the fly larva moves about more than average. Sokolowski found that the allele frequencies in a natural population of Drosophila melanogaster are 70% for to the power R and 30% for to the power S. She noted, however, that while the mechanistic or machinistic basis for the differences in rover and sitter behaviors are well known, little is known about the evolutionary and ecological significance of different frequencies of these alleles in natural populations. Sokolowski teamed with Sofia Pereira and Kimberly Hoggs of York University to conduct a laboratory study of how population density might affect the frequency of the 4 to the power R and 4 to the power S alleles in Drosophila populations. Sokolowski and her colleagues raised Drosophila at both high and low densities, starting with equal frequencies of 4 to the power R and 4 to the power S in both lineages. After 74 generations, larvae from the low density and high density lineages showed a clear divergence in behavior as measured by differences in average foraging path length, as shown this figure or this graph. The researchers conclude that the 4 to the power S allele had increased in frequency in the low density populations in which short distance foraging would yield sufficient food, while long distance foraging would result in unnecessary energy expenditure. Meanwhile, the 4 to the power R allele had increased in frequency in the high density range, where long distance foraging could enable larvae to move beyond areas of food depletion. In other words, they had observed an evolutionary change in behavior in their laboratory populations. Migratory patterns in black caps. An example of evolutionary change in the behavior of white population comes from studies on migratory behavior in a population of black caps. The migratory patterns of black caps are well known in Western Europe because of a great interest in bird study and a long history of bird banding. Black caps that breed in northwestern Europe generally migrate to the western Mediterranean region for the winter. As the consequence, black caps in Germany have historically migrated in a southwest direction, while those in Britain have migrated south. In the 1950 years, a few black caps began to spend their winters in Britain, and over time the population of black caps wintering in Britain has grown to many thousands. Surprisingly, these wintering birds are not birds that bred in Britain and then stayed for the winter. Lick bands on some of the birds show that they had come from continental Europe and migrated westward in Britain instead of south to the Mediterranean. Peter Brithold and his colleagues captured 20 male and female black caps wintering in Britain and transported them back to their laboratory in southwestern Germany. They compared the migratory orientation of these birds of their F1 offspring to to that of young bears originally from southwestern Germany. Behavioral ecologists have long known that the migratory restlessness that Berthold has studied previously is not a random activity, but rather is directed. That is, caged bears showing migratory restlessness orient their activity in the direction that they would migrate if they were not caged in the autumn. When the bears were showing migratory restlessness, Berthold's team placed the black cabs from their three study groups in large glass-covered funnel cages lined with carbon-coated paper for 1.5 to 2 hours. As the bears moved around the funnels, the marks they made on the paper indicated the direction in which they were trying to migrate, as shown this figure. The results showed that the migratory orientation of wintering adult bears captured in Britain was very similar to the orientation of their laboratory raised offspring. That orientation contrasted sharply with the migratory orientation of the young bears originally from Germany. This study indicates a genetic basis for migratory orientation, since the young of the British black caps and the young bears from Germany were raised under similar conditions but showed very different migratory orientations. But has the behavior actually evolved over time? Berthold's study also indicates that the change in migratory behavior in Western Europe, black caps in both recent and rapid, having taken place over the past few decades. Before 1960, there were no known westward migrating black caps in Germany, but by 1990, westward migrants made up 7 to 11% of the black cap populations of Germany and parts of Australia. Berthold suggests that once westward migration began, it persisted and increased in frequency as a result of several factors, including milder winter climate in Britain and improved winter food, partly due to widespread use of winter bird feeders in Britain. Notice the directions wintering black caps captured in Britain and their laboratory raised offspring had a migratory orientation toward the west, while young bears from Germany were oriented toward the southwest. Notice the direction. Natural selection favors behaviors that increase survival and reproductive success. The genetic components of behavior, like all aspects of phenotype, evolve through natural selection for traits that enhance survival and reproductive success in a population. Two of the most direct ways a behavior can affect fitness and through its influences on foraging and mate choice behavior.